Good morning children. Welcome back to our botany class today. Children, I hope you all completed the previous homework what I have given already. Is it? Okay children, in the previous class we all learned about the dihybrid cross, Mendel's law and also about the structure of chromosome. Isn't it now? What about the phenotypic ratio of dihybrid cross? 9:3:3:1. What are the Mendel's law? First law of dominance. Second is law of independent, law of segregation. Third is law of independent assortment, isn't it? Okay, then you all learned about the structure of chromosomes also. What are chromosomes? Thin, linear structures present in the nucleus of the cell, that is called chromosomes, isn't it now? So chromosomes have a central body that is called centromere. You all know the structure of chromosomes, isn't it? Chromosomes have a central body that is centromere and have the arms. These arms are chromatids. The chromatids are attached by a central body or it is called centromere, isn't it? Today we are going to learn about the types of chromosome based on the position of the centromere and the types of chromosome based on their function. Okay children, the first is about the types of chromosomes based on the position of the centromere. What is centromere? The central body, isn't it? Or the chromatids are joined with the help of a central body or it is called centromere. There are 4 types of chromosomes based on the position of the centromere. Okay ma'am? What are the 4 types of chromosomes based on the centromere? One is telocentric, okay? Telocentric, acrocentric, metacentric and submetacentric, okay? Submetacentric. These are the types of chromosomes based on the position of the centromere. First one is telocentric. Telocentric chromosomes. The centromere, this is the telocentric chromosome. The centromere is found at the proximal end, okay? Or at the terminal part, okay? The centromere is present at the proximal end. These chromosomes are rod shaped, okay ma'am? Next is acrocentric. Acrocentric chromosome. This is acrocentric chromosomes. These are the chromatids, okay? What is this? Centromere. Centromere. This is centromere. These are the chromatids. Okay. Now this acrocentric, this chromosomes, centromere is present at one end with the long arms and a short arm. Okay. This centromere is present. Here this chromosomes having long arms and short arms. Okay, ma'am. These chromosomes have the short and long arms. Centromere is here. Next, what is this metacentric chromosome? The metacentric chromosome, the centromere is present in the center of the chromatids. Centromere is present near the center of the chromosome. These arms shows so these 2 arms are equal, isn't it? These 2 arms are equal. So metacentric chromosome, centromere occurs in the center of the chromosome and form 2 equal arms. These 2 are equal. So it is metacentric. The chromosomes are V-shaped chromosomes. Metacentric chromosomes are V-shaped chromosomes. Okay. Next, submetacentric chromosome. Centromere is present almost near the center. Centromere is present near the center of the chromosome, but the chromosomes have unequal arms. These chromosomes are L-shaped or J-shaped. Okay, L-shaped or J-shaped. These chromosomes are L-shaped or J-shaped. Okay, so these are the types of chromosomes based on the position of the centromere. Next is the types of chromosomes based on their function. Okay, based on their function, the chromosomes are of 2 types. They are allosomes and autosomes. Okay, there are 2 types of chromosomes based on the position of the centromere: allosomes and autosomes. Eukaryotic chromosomes are classified into autosomes and allosomes. What are eukaryotes? The organisms having, the cells having membrane bound organelles. They are called eukaryotic cells. Okay. So, the cells having membrane bound organelles. Membrane bound organelles in the sense, nucleus is surrounded by membrane. Each and every organelle such as mitochondria, ribosomes, lysosome, all these membranes are bounded by organelles. These membrane bound organelles are present in eukaryotic cells. Eukaryotic chromosomes are classified into allosomes and autosomes. Okay, autosomes contains the genes that determines the somatic characters. Somatic means body. Okay, body characters. The chromosomes which determine the body characters are autosomes. So chromosomes which will determine the body characters are in the sense somatic characters. These are called what? Autosomes. Okay. Then this allosomes. Allosomes are the sex chromosomes, or it is termed as heterochromosomes. These are sex chromosomes. Allosomes which determines the sex of an organism. Okay, ma? So what about autosomes? Autosomes contains the genes that determine the somatic characters. Male and female all have the same or equal number of autosomes in their body. It is also called somatic chromosomes. Male and female have the same or equal number of autosomes. Okay. Then allosomes are otherwise termed as sex chromosomes or heterochromosomes. Allosomes are also called sex chromosomes or heterochromosomes. There are two types of sex chromosomes. They are X and Y chromosomes. There are two types of allosomes or sex chromosomes. What are they? X and Y. Female have two X chromosomes. Female have XX, two X chromosomes, and male have one X and one Y chromosomes. Okay, this is present in female. This is present in males. Okay, ma'am. So human male have one X and one Y chromosome. Human male have one X and one Y chromosome. Human female have two X chromosomes. These are allosomes. Okay. So based on the position of the centromere, chromosomes are 4 times. Based on their function, chromosomes are 2 types, isn't it? Autosomes and allosomes. Now, next about the karyotype. Karyo means nucleus, isn't it? Karyotype. Then karyotype. Karyotype. Every organism have constant or a particular number of chromosomes. In human, each cell normally contains 23 pairs of chromosomes. Okay, how many chromosomes are there in our body? 23 pairs. Out of 23 pairs, 22 pairs are autosomes. And 23rd pair is the sex chromosomes or heterochromosomes. Okay, karyotype. Every organism have a constant number of chromosomes. In human cell normally contains 23 pairs of chromosomes, out of which 22 pairs determines the body character. They are called autosomes, and the 23rd pair determine the sex of an organism. They are called allosomes or sex chromosomes. So the body cells of sexually reproducing organisms, the chromosomes generally, body cells or autosomes generally occurs in pairs. That is called a diploid. Okay, diploid, it is designated as 2n. Sexually reproducing organisms, the chromosomes are generally occurs in pairs said to be diploid. These organisms produce the gametes, isn't it? Male produce the male gamete and female produce the female gamete. So the gametes produced by these organisms contain a single set of chromosome that is said to be haploid. Okay, that is said to be haploid. It is designated as N, haploid N. Okay, so the gametes produced by the organisms contain a single set of chromosome that is called haploid. So karyotype is the size, shape and number of chromosomes in the cell nucleus of an organism. Karyotype is the number, size and shape of chromosomes in the cell nucleus of an organism. Okay, now this next is about the structure, this ideogram. It's a diagrammatic representation of the karyotype of a species, that is ideogram. Ideogram is the diagrammatic representation of what? This karyotype of a species. You know what is karyotype, isn't it? Shape, number, and karyotype is the shape, number, and position of a chromosomes, isn't it? So what is ideogram? Is a diagrammatic representation of karyotype of a species. You all know that the metaphase, during metaphase, in cell division during metaphase, or the homologous chromosomes lie at the equatorial plane, isn't it? There, the length, thickness and position of centromere, shape, etc. with the sex chromosome placed at the homologous pairs according to decreasing the length, thickness, position of the centromere, shape, etc. the sex chromosome placed at the end. Okay. Now the structure of DNA. As you all know about DNA, isn't it? It's a hereditary material. This hereditary material, it contains the genetic information, isn't it? It contains this structure of DNA. Next is the structure of DNA. It's a hereditary material. It contains the genetic information. The most widely accepted model of DNA is the double helical structure proposed by James Watson and Francis Crick. James Watson and Francis Crick. Okay, their model is the most accepted model of DNA. They propose the double helical structure of DNA. Okay, they propose the three-dimensional structure of DNA based on the X-ray diffraction studies of DNA obtained by Rosalind Franklin and Morris Wilkins. For their discoveries, they awarded Nobel Prize. Okay, so what about DNA? DNA is the hereditary material. It contains the genetic information. It is the most important constituent of the chromosome, isn't it, ma'am? Then the chemical composition of the DNA molecule. The DNA is a here, this is the structure of DNA. DNA is the large molecule. It is consisting of millions of nucleotides. So it is called polynucleotide. Okay. So why we call them as why we call the DNA as polynucleotide chain? Because it consists of millions of nucleotides. Hence it is called polynucleotide. Each nucleotide consists of 3 components. Okay. Each nucleotide consists of how many components? 3 components. A sugar molecule, a nitrogenous base and a phosphate group. Sugar molecule, nitrogenous base and phosphate group. Okay. Phosphate group. These are the 3 important constituents of DNA. So the model of DNA was proposed by James Watson and Francis Crick based on the X-ray diffraction studies made by Franklin Wilkins and Morris and the other scientists, namely this Wilkins and Morris Wilkins, isn't it? Now, for their discoveries, they awarded Nobel Prize. The DNA is a polynucleotide chain consisting of millions of nucleotides. Then it consists of three important components, namely, what are they? Sugar, nitrogenous base, and a phosphate group. What are they now? Sugar molecule, nitrogenous base, and phosphate group. This nitrogenous base found in DNA are of two types. What are they? Purines and pyrimidines. Okay, purines and pyrimidines. This nitrogenous base found in DNA are of two types, namely purines and pyrimidines. Okay, mom. These purines are adenine and guanine. Purine bases are adenine and guanine. These two are the purine bases. And pyrimidine bases are thymine and cytosine. Thymine and cytosine, these two are the pyrimidine bases. Okay, so nitrogenous bases, so there are three important constituents found in DNA. What are they? Sugar, nitrogenous base, and phosphate group. Nitrogenous bases found in DNA are of two types, purines and pyrimidines. These purine bases are adenine and guanine. Pyrimidine bases are thymine and cytosine. What are the purine bases? Adenine and guanine. What are the pyrimidine bases? Thymine and cytosine. These are the pyrimidine bases. Now, you must know what is nucleotide and what is nucleoside. Okay, this nitrogenous base combines with sugar molecule to form nucleoside. Okay, nitrogenous base combines with sugar to form nucleoside. What is nucleoside? Okay, ma'am. Then what is nucleoside? The side is nucleoside plus nucleoside combines with the phosphate group, to form nucleotide. Okay, DNA consists of millions of nucleotides, so it is termed as polynucleotide chain, isn't it? What is called nucleotide? Nucleoside combines with the phosphate group to form nucleotide. What is nucleoside? Nucleoside is nitrogenous base combines with the sugar to form nucleoside. Okay, so nucleoside and nucleotide, you must know the difference between nucleotide and nucleoside. What is nucleoside? Nitrogenous base. What are the nitrogenous bases? Purines and pyrimidines. Name the purine bases: adenine and guanine. Name the pyrimidine bases: thymine and cytosine. Okay, so this nitrogenous base combines with the sugar molecule to form nucleoside, and this nucleoside combines with the phosphate group to form nucleotide. Okay, so this nucleotide and nucleoside. This Watson and Crick model of DNA. The DNA molecule consists of polynucleotide chains. What are called polynucleotide chain? The DNA consists of millions of nucleotides. What are nucleotides? Nucleoside combines together with phosphate to form nucleotide, isn't it? The nucleotides are formed according to purines and pyrimidine bases present in the DNA. Okay, so DNA molecule consists of polynucleotide chain. These chains form a double helical structure. It's a polynucleotide chain. This is one helix, this is another helix. So this DNA form a double helical structure. Okay, these chains form a double helical structure, and these two strands which run anti-parallel to each other. Now this these nitrogenous base in the bases found in this is the structure. This is also a structure. The nitrogenous bases found in the center. This is nitrogenous base. What is it? Thymine, adenine. cytosine, guanine, sugar and phosphate molecule. Okay, sugar, phosphate molecule, these are the nitrogenous bases. So, nitrogenous bases in the center are linked to sugar phosphate units which forms the backbone of the DNA. Okay, nitrogenous base found in the center or linked to sugar phosphate. Okay, this is found in the center. Nitrogenous base is found in the center linked to the sugar phosphate molecule. These forms the backbone of the DNA. Then pairing between nitrogenous bases specific or pairing of nitrogenous bases specific, this adenine always pairs with thymine with the 2 hydrogen bonds. Okay, this base, the base pairing is specific. You know that this purine and pyrimidine bases, isn't it? Pyrimidine bases are cytosine and what are purine bases? Adenine, guanine. This pyrimidine bases are cytosine and thymine. Okay, cytosine and thymine. Now this base pairing is specific, isn't it? Pairing between nitrogenous base is specific between purine and pyrimidine. So what are the purine bases? Adenine, guanine. Pyrimidine bases thymine and cytosine, isn't it, ma'am? Then this adenine always pairs with thymine with the 2 bonds, with the 2 hydrogen bonds. Adenine always pairs with thymine with the 2 hydrogen bonds. Guanine always pairs with cytosine with the 3 hydrogen bonds. Listen here, adenine always pairs with thymine with the 2 hydrogen. One of the purine base pairs with one of the pyrimidine base. Okay, what base pairing is specific. You all know that base pairing is specific. One of the purine base linked with one of the pyrimidine base. Adenine always pairs with thymine and guanine always pairs with cytosine. Adenine pairs with thymine with the 2 hydrogen bonds. Guanine always pairs with cytosine with 3 hydrogen bonds. Okay, so hydrogen bonds between nitrogenous base made the DNA stable. Okay, hydrogen bonds between the nitrogenous base. These are the hydrogen bonds between. So hydrogen bonds between the nitrogenous base make the DNA stable. Okay, ma. Each turn of double helix is 34 angstrom. Okay, or 3.4 nm, or it is 34 angstrom. Okay, ma. This each turn of double helix is 34 angstrom. There are 10 base pairs in each turn. Okay, there are 10 base pairs. Base pairs in the sense adenine always pairs with thymine, guanine pairs with cytosine. This base pair, there are 10 base pairs in each turn. So in a complete turn, how many base pairs will be there? 10 base pairs. Okay, so nucleotides in a helix are joined together by a phosphodiester bonds. Okay, ma'am. So nucleotides are linked together by a phosphodiester bonds. This is the structure of DNA. So structure of DNA, the model is proposed by Watson and Crick. Okay, the model of DNA is proposed by Watson and Crick. James Watson and Francis Crick. Okay, this is the model of Watson and Crick. Model of DNA. DNA molecule consists of how many nucleotides? 2 polynucleotides. Why it is termed as polynucleotide? Because it consists of millions of nucleotides. What are called nucleotides? Nucleoside combines with the phosphate group to form nucleotides. Okay. Now this what is called nucleoside? Nitrogenous base combines the sugar molecule to form nucleoside. Okay, so what are the 3 important constituents of DNA? What are the 3 important constituents? Nitrogenous base, sugar and phosphate. Okay, what are the 2 nitrogenous bases found in DNA? Purines and pyrimidines. Name the purine bases: adenine and guanine. pyrimidine bases, cytosine and thymine. Okay, so base pairs are specific. That means adenine always pairs with thymine. Guanine always pairs with cytosine. The bond between the hydrogen bonds between adenine and thymine is double bond, and guanine and cytosine is triple bond. Okay, all these nucleotides are linked together by a phosphodiester bonds. This is the structure of DNA. Okay, children, now you all turn to page 271. Okay, children, you all turn to page 271. So choose the correct answer, not the answers. Choose the correct answer. Fourth one, the centromere is found at the center of the dash chromosome. What is it marked? Metacentric. The next, the fifth question, the dash units form the backbone of the DNA. Nitrogenous bases. Nitrogenous bases. Next one, next fill in the blanks. Here also one more seventh question. Seventh question, the number of chromosomes found in human being are 22 pairs of autosomes and 1 pair of allosome. Allosome means sex chromosome. Fill in the blanks. Fourth one, DNA consists of 2 dash chains, polynucleotide chains. Polynucleotide. What is it right? Polynucleotide. Polynucleotide chains. Next one. Turn next page 272. Match the following autosomes. Autosomes, 22 pairs of chromosomes. Diploid condition, 2n. Diploid condition, 2n. Allosomes, 23rd pair of chromosome. Down syndrome, that is trisomy 21. Dihybrid ratio 9:3:3:1. Okay. Then short answer question. Question number 3. What are allosomes? Answer is there in page 266. Answer is there in page 266. What are allosomes? Last paragraph, first part, last paragraph, up to heterochromosomes you mark it. Allosomes are chromosomes which are responsible for determining the sex of an individual. They are also called sex chromosomes or heterochromosomes. Up to that you mark it. Then long answer question. Explain with an example the inheritance. Second one, how is the structure of DNA organized? Answer is there in 267. 267 and 266. Starts from here. The structure of DNA, Roman number 7-2. DNA is the hereditary material. Up to the 7th point, nucleotides in the helix are joined together by phosphodiester bond. Okay, phosphodiester bond. Significance of DNA, biological significance of DNA, you can mark it in the next class. Children, you have to learn and write these answers. Short answer, Roman number 6, question number 3. Then Roman number 7, question number 2. Roman number 7, question number 2. That is the long answer. You are learning and writing in the notebook. Then choose the correct answer. Choose the correct answer 4 to 7. You are learning and writing in the notebook. Okay. Short answer 1. Then short answer 1. Long answer 1. That is Roman number 7-2. You are learning and writing in the notebook. Then fill in the blanks. What we marked today. These are the answers you are writing in the learning and writing in the notebook. Okay. Thank you, children. We shall meet again in the next class.